inviting me to give grand rounds today. I'm excited to get to share some of this work with you all. So I'm going to talk today about the potential for germline genetics to improve risk-stratified screening for prostate cancer, and I have no disclosures to report. So the three main objectives that I really hope that you take away from this talk today are to be able to recognize that germline genetic risk factors for prostate cancer are not diagnostic, but they could potentially inform future risk of disease development. Also, I hope that you'll gain an appreciation of how genetics could be used to inform when to initiate prostate cancer screening and how frequently screening should occur. And last, that you'll be able to identify some genetic risk factors that increase risk of overall and aggressive prostate cancer. So to give you a little bit of background first, prostate cancer represents one of the biggest health disparities in the US today. The incidence rates in Black men are about 1.5 to 1.75 times higher than they are in white men, as it's shown in this figure here. Let me see if I can get it. And this is relative to white men, which is represented by the black horizontal line. But you can see when we look at mortality rates, an even more severe disparity, where Black men have had mortality rates that are 2 to 2.5 times higher than white men over the last 2 decades, and this remains true today. Despite being a very common cancer, there's very few established risk factors for prostate cancer. Some of those include age, population, as shown in the figures here, and family history or germline genetics, with the heritability of prostate cancer being about 58%. And this means that this is actually one of the most heritable prostate cancers, so there's a very strong genetic component to prostate cancer. And when we look at the genetic studies that have been done, as of 2 years ago at least, we can see that there are a number of genetic risk variants across the genome that are associated with prostate cancer risk. So this figure here is a karyogram, and it's showing each of the vertical lines is one chromosome of the genome, and the little dashes next to each chromosome represent a region of that chromosome that has been associated with prostate cancer risk. And this is looking at both overall prostate cancer risk and more aggressive prostate cancer too. And something that this also shows that's kind of hard to see by looking at all the little dots is that rare as opposed to common variants have had more evidence of being associated with risk of aggressive prostate cancer. So some of these genes with rare variants include BRCA2 and ATM, which you may have heard of. Despite prostate cancer having such a big disparity and having a really strong genetic component, genetic studies to date have had a lack of diversity that could potentially contribute to health disparities. Consequently, when we develop polygenic models of different traits or diseases, we tend to see predictive accuracy. Let me see if I can find the little mouse. That's increasingly poor in populations that are more ancestrally distant from European ancestry. And in particular, we often see pretty poor performance in individuals of African ancestry. And you can imagine that in conjunction with existing health disparities, for instance, what we see in prostate cancer, that if we use polygenic models that have poor predictive accuracy or predictive ability in the populations most burdened by these conditions, that it could potentially exacerbate existing health disparities. So this is something we've been pretty aware of in prostate cancer, and we've been trying in recent years to really try to overcome this problem by including as many diverse participants as we can in our genetic studies. So I'm going to talk predominantly about 3 different topics today. I'll talk about work that we've done to construct a multi-ancestry polygenic risk score, or PRS for short. I'll talk about some population-specific genetic risk factors, and then we'll also talk about work we've done to combine polygenic risk scores with rare genetic variants. And the main objective that I want you to take away is that genetic risk factors inform future risk of prostate cancer across diverse populations that could potentially improve risk-stratified prostate cancer screening and reduce health disparities. So first, I'll talk about polygenic risk scores. So we conducted a couple years ago a large and diverse prostate cancer genome-wide association study, or a GWAS study. So in this study, we had over 230,000 men across the population shown in this table here. And what's shown in this figure is called a Manhattan plot. So on the x-axis, we have the different chromosomes of the genome. On the y-axis, we have the negative log p-value. And each dot represents one association between a genetic variant and prostate cancer risk. So the higher the dot, the stronger the association with prostate cancer risk. And you can see here that we tested about 17 million variants, one at a time, for association with prostate cancer risk. After running this GWAS, we found that there were a total of 169 variants that were strongly associated with prostate cancer risk. And in aggregate, these account for about 34 to about 43% of the familial relative risk of prostate cancer, meaning that of the variation of prostate cancer that we know can be attributed to genetics, we're able to account for about 30 to 40% of it, which is pretty good. We're accounting for a lot of the genetic variation, but there's still about 60 or so percent remaining that we haven't yet accounted for that we should be able to with genetics. So we took these 269 variants and used them to construct a polygenic risk score, which is just a weighted sum of the number of risk alleles carried by each participant. So in this equation here, it's a really pretty simple one. I won't show a lot of equations, but you just sum up the number of risk alleles carried by an individual for a given variant, and you weight that variant, the number of risk alleles, by a multi-ancestry weight. And then you get one single score for each participant, and that score represents their inherited risk for prostate cancer, just based on these common 269 variants. And this is a multi-ancestry polygenic risk score, so we use the same equation for any participant regardless of what population they come from. So once we have this polygenic risk score calculated, we can look at the distribution of the polygenic risk score in cases and controls as shown in this figure here. And you can see that in cases, the PRS distribution is a little bit higher than it is in controls, which we expect to see if the PRS is working as we would expect it to. So once we have the PRS calculated, we like to evaluate the association with prostate cancer risk by stratifying all of our participants into deciles. And those decile cutoffs are determined by the distribution of the PRS in controls, as shown in this figure here. We then calculate the number of cases and controls that fall into each PRS category. For instance, how many cases and controls are in this top PRS decile versus how many cases and controls are in this average 40 to 60% category. And we can then calculate an odds ratio for each of the PRS deciles by making those comparisons. So we did this in a large sample of over 500,000 men who were independent from our GWAS, so they were not included in the discovery where we constructed the polygenic risk score. Most of these, a big bulk of them, were from the Million Veteran Program, but we also had 5 other really large biobanks and other studies that were contributing to this PRS validation. And here I'm just showing the odds ratio of prostate cancer in each PRS category, again relative to the average 40 to 60% category. But you can see if you compare these odds ratios between studies and populations, they're pretty similar. We see pretty good replication across populations and across studies, which is promising. If we average those across the different populations, you can see that the odds of prostate cancer range from about 2.8 in African ancestry men, shown by the blue dot, up to about 3.8 in European ancestry men, shown by the pink dot. So this is a pretty big odds ratio. We're showing that men with high polygenic risk scores have really high odds of prostate cancer compared to men with average genetic risk. So it's capturing a lot of potential variation in prostate cancer risk and could be really clinically informative. We do see the smaller odds ratio in African ancestry men. It is doing pretty well across populations. There's still a lot of information to be gained, but we still have room for improvement to bring that odds ratio up because we know that African ancestry men have higher risk, so we probably would expect that to be a little bit higher if it was doing as good as it could be. There's a few other ways that we evaluated the polygenic risk score. We stratified participants by age to see if the effect of the polygenic risk score on prostate cancer risk varies by age, and what we ended up seeing was that in younger men, shown by the left in each of these figures, there's a stronger association or a bigger association with prostate cancer risk associated with the PRS in younger men. And in older men, that association gets a little bit weaker. This is true in both European ancestry men and in African ancestry men. So this is suggesting that the polygenic risk score would likely be most informative of prostate cancer risk in younger men, which makes sense because the older you are, the more time you've had to accumulate environmental exposures that could impact your risk of prostate cancer. Whereas if a younger man is diagnosed with prostate cancer, there might be a stronger genetic contribution to that, to his prostate cancer. And then similarly, sorry, this figure didn't turn out looking too good, but we also saw, if we look just within men who have prostate cancer, that men with a higher polygenic risk score, shown by the x-axis here, tend to have a younger age at diagnosis. So this is looking across many different populations, and we see this pretty consistently. But if you just look, for instance, within East Asian men, you can see that the higher the PRS, the younger the mean age at diagnosis. And it looks like there are differences between mean PRS, age at diagnosis. It looks like there's differences of age at diagnosis between populations, but that's really just sampling strategies, like differences in the study design, rather than actual differences in age at diagnosis. And then one more thing that we looked at was the absolute risk of prostate cancer by age and PRS category. So here in these figures, we're showing the absolute risk of prostate cancer on the y-axis. We're looking at age on the x-axis, and the different colored lines represent absolute risks for different PRS categories, with the higher lines representing higher PRS categories. So you can see that the bigger the PRS, the higher the PRS, the higher the absolute risk of prostate cancer. And this is really consistent across European, African, and Hispanic populations. And in particular, we also know if we just look at this 5% absolute risk threshold, that men who are in the highest PRS decile, looking at this orange line here, on average reach this 5% absolute risk threshold 12 years younger than men who have average polygenic risk scores. In African ancestry men, that difference is 8 years, and in Hispanic men, it's about 12 years. So this is really suggesting that the polygenic risk score could likely inform at what age would be ideal to start screening for prostate cancer. And then likewise, when we look at these absolute risk curves, integrating family history into the picture, you can see that there's a lot of additional information to be gained. Similar to the last figure, we see that the higher the PRS, the higher absolute risk. But now we also see that high PRS and high family history leads to an even higher absolute risk score, which of course makes sense, but it just kind of goes to show that looking at this information in conjunction is likely much more informative than just looking at one or the other alone. An important question to ask in prostate cancer is whether you can distinguish aggressive prostate cancer risk from non-aggressive prostate cancer risk. And when we look at how our aggressive prostate cancer cases are distributed across PRS categories, which is shown the PRS categories here on the y-axis, you can see that about 50% of the aggressive cases, represented by the brighter color in this figure, fall into the top PRS quintile, so the 80 to 100% PRS category. And this is for European ancestry men, but for African ancestry men, we similarly see about 45% of aggressive cases falling into that top PRS quintile. So this is suggesting that if we were to use the PRS in a clinical setting, we probably would capture a large percentage of the aggressive cases. To more directly ask this question, though, we also look to see how do associations differ between different prostate cancer statuses. So in this figure, we're showing the odds ratio for different prostate cancer outcomes versus controls. And in particular, if we compare risk of non-aggressive prostate cancer versus controls and risk of aggressive prostate cancer versus controls, you can see that there's a significantly larger risk of aggressive as opposed to non-aggressive disease. But to more directly address this question, we also looked at the odds ratio for metastatic versus non-aggressive disease. And for men in the top PRS decile, we see that they have about 65% increased risk of metastatic versus non-aggressive prostate cancer when we compare them to men with just average genetic risk. And this is something that we importantly have only seen in this particular study and others, but this is a really large sample of African ancestry men, and we haven't seen this as convincingly in other populations. But we see it pretty consistently in African ancestry men, which is suggesting that the PRS could potentially be a way to identify African ancestry men who might have increased risk of aggressive disease. So this study was published last year where they asked the question of, is the polygenic risk score associated with converting from active surveillance to treatment for men who are initially diagnosed with really indolent prostate cancer, and they and their clinician decide that active surveillance will be a good route of treatment for them or a good course for them. But you can see here that men who had higher PRS tended to be a little bit more likely to convert from active surveillance to treatment, which could suggest that maybe these men were upgrading and that's why they switched to treatment. But there's a lot of reasons why you could switch to treatment, including just having anxiety about continuing active surveillance. So we wanted to more directly ask this question, and we looked at this in the Canary Pass study. This is still ongoing work right now that we're doing. But here we have about 1,200 men who are on active surveillance, and 470 of them classified or upgraded to a higher grade of prostate cancer while they were on active surveillance. And we included all of the diverse participants we could, which is it's still 85% non-Hispanic white men that are included in this study, but we tried to conduct an inclusive analysis so that we would be able to include all of the diverse participants that we have in PASS. But importantly, we saw that for each standard deviation unit increase in the polygenic risk score, men had 27% higher risk of grade reclassification, which is it was really statistically significant and a reasonable increase in percent risk. In this study, we also saw that higher PRS was associated with a smaller prostate size, higher PSA density, and a higher percentage of positive biopsy cores at diagnosis. So these are all factors that could suggest risk of more aggressive disease. So to summarize the improvement that we saw with this polygenic risk score that was constructed from a diverse population, which is represented by the 2021 row here. In 2018, we had a GWAS that was conducted largely in European ancestry men. And from that, there were about 180 variants that were used to construct a polygenic risk score. But now with the addition of about 30,000 non-European ancestry cases and a large number of non-European ancestry controls, we have these 269 variants in a new PRS. And you can see that the odds ratios we're able to evaluate now or assess with this new PRS are much larger than what we previously were able to get. So we do see a lot of improvement with this new PRS that incorporates diverse populations. And more recently, we just had a paper that was accepted, and I'm a co-author on this, so I'm not going to talk about it too much, but this was accepted in Nature Genetics, where we did a bigger, diverse GWAS study that had over 900,000 men with more diversity than we previously had. And this led to the identification of 451 variants, which we use to construct a polygenic risk score, and we see a little bit of improvement in the polygenic risk score with this new GWAS that was just completed. So one other question that we wanted to address with regards to polygenic risk scores is thinking about these genome-wide polygenic risk scores. So there's been a lot of recent studies that have shown that these genome-wide approaches to constructing a polygenic risk score, where you choose variants across the genome, it could be millions of variants, regardless of their association with disease risk, often lead to a polygenic risk score that is more strongly predictive of disease risk than just choosing the variants that are significantly associated with disease risk. This has been seen for diabetes and for cardiovascular disease pretty consistently. So we wanted to see if our polygenic risk score of 269 variants was doing better or similarly to these genome-wide approaches. So we addressed this in this study where we constructed we took these 6 different algorithms to constructing a genome-wide polygenic risk score, and the names of the algorithms are shown in bold here. Once we had them constructed, using the same summary stats that we used to develop the PRS for 269 variants, so it's kind of a fair comparison. We compared these genome-wide approaches to our 269 PRS using independent data, testing data of African ancestry and European ancestry men, and then we did additional validation in an independent sample of African and European ancestry men from the Million Veteran Program. Once these PRS were calculated in these independent data sets, we then evaluated the performance by looking at the area under the curve, or AUC, and then looking at the odds of prostate cancer associated with the PRS. So I'll just jump ahead to the validation results, looking in African ancestry men. So here in red, let me see if I can get the mouse to show up again. This is showing the AUC for a covariate-only model, where we just have age and then principal components of ancestry, so genetic ancestry in this model. You can see that when we add to this model our 269 PRS, we see a really huge improvement in AUC. We also see improvement in AUC if we just add each of the genome-wide PRS approaches, which are shown by the different colors, but it's not nearly as big as what we see for our 269. And our 269 really has significantly better performance than any of the genome-wide approaches that we evaluated. Similarly, when we look at odds of prostate cancer, you see that we have a larger odds of prostate cancer for our 269 PRS compared to any of the genome-wide approaches that we looked at. When we look in our replication sample of European ancestry men, we see pretty similar pictures as what we previously observed. Our 269 PRS has the best AUC and the best odds of prostate cancer, the largest, suggesting that it has the best performance. But now we also see that this particular genome-wide approach, shown by yellow, which is called PRS-CSX, has pretty good performance. It's similar. It's not better, but it's similar to what we see for our 269. But that includes 1.1 million variants as opposed to just 269 variants. So if you're thinking about clinically implementing something like this, it would be much simpler to just do 269 variants where we have some insights about the potential biological implications of these variants. So to date, it seems that these genome-wide approaches do not have better performance than our simpler, more carefully created polygenic risk score, but this probably will be something we'll have to reassess in the future as new methods come out. So to summarize this first section, could PRS improve risk-stratified prostate cancer screening? So we saw that men in the top PRS decile reached this 5% absolute risk threshold about 8 to 12 years younger than men in an average PRS category, which could suggest that the PRS may inform at what age men should start screening and how frequently they should screen. We also saw that men in the top PRS decile have 65% increased risk of metastatic versus non-aggressive prostate cancer. And this is something we only saw in African ancestry men, but it could suggest that the PRS may be able to identify African ancestry men with increased risk of lethal disease. But ultimately, we'll need much larger sample sizes of non-European ancestry men to further improve the performance of the polygenic risk score across populations. So in this second section, I'll talk about some population-specific genetic risk variants that we've identified. So this, first, I'll just give two examples. This first one was based on a paper that was published a few years ago, where they conducted a GWAS in a large sample of African ancestry men, and they have this really strong peak that you can see here in chromosome 8. The strongest signal was this variant in the 8q24 region that I'll refer to as RS727 for short. It's a fairly common variant. About 11% of African ancestry men carry this variant, and it has a really large odds ratio of 2.3. Usually in GWAS studies, the variants we identify have odds ratios of maybe 1.1 or 1.2, so this is a really big odds ratio for a single variant. This particular variant is in a non-coding intergenic region near prostate cancer associated long non-coding RNAs, but it lies in a prostate cancer-specific enhancer. So there, even though it's a non-coding variant, there's some potential biological function for this variant. So we wanted to follow up on this particular variant to better understand it. So we found that this variant is more common in men who have a stronger family history of prostate cancer and those who are diagnosed at a younger age. We also found that the risk allele is increasingly common in men who have more aggressive disease, with the odds ratio being up to 2.7 for men who carry one copy of this variant when we're looking at risk of lethal prostate cancer. This variant alone accounts for about 9% of the familial relative risk of prostate cancer, and to give you a sense of how much that is, at the time there were 180 known variants, and those account for about 28% of the familial relative risk. So this one variant alone is counting for about a third of the variation that we could account for with genetics at the time that we did this. So that's really a pretty huge percentage. And importantly, when we look at the genetic ancestry of participants, we can see that the risk allele frequency is much more common in men who have a higher proportion of African ancestry. So this is just going to show that this pretty important risk factor that we've identified wouldn't have been able to be identified if we only had European ancestry men contributing to our GWAS study. So this is a really interesting finding that we're only able to identify because we are able to include diverse participants in our studies. And then the last thing I want to mention for this variant is that when we looked at the absolute risk of this variant by age, carrier status, so here age is on the x-axis, carrier status is represented by the different colors of the lines here. And then we also looked by family history, and family history positive rates are shown in the dotted lines. Family history negative is the solid line. So if we focus just first on the family history positive rates, you can see that men who carry 0 copies of the risk allele who are family history positive have a 9% absolute risk of prostate cancer by age 60. That increases to 21% in men who carry just 1 copy of this allele and are family history positive, and up to 38% in men who carry 2 copies. 38% absolute risk of prostate cancer by age 60 is a pretty big percentage. So this is really suggesting that this variant alone could be very informative for risk-stratified screening. The highest absolute risk that we observed reaches about 80% by age 85 in homozygote carriers, men who carry 2 copies of the allele, who are also family history positive. And then I just want to also point out that the absolute risk in men who are family history negative, who carry 1 risk allele, is about the same as men who are family history positive and carry no risk alleles. So this is really conveying a lot of additional information beyond family history and could be used in a complementary way to family history. So this seems like it's a really important risk factor for prostate cancer in African ancestry men, and what are the ways that we could incorporate this into screening? I think one good example of this is looking at the European ancestry specific HOXB13 variant, which is referred to as G84E. That variant accounts for about 5% of the familial relative risk, as opposed to the 9% that we see for our RS727 variant. And men who carry that variant are recommended to consider screening starting at age 40 or 10 years before the youngest diagnosis in their family. If we had similar guidelines for this 8Q24 variant that is only found in African ancestry men, that could potentially prove beneficial for prostate cancer screening. The second example I'm going to talk about is digging a little bit more into HOXB13 that we just mentioned. So HOXB13 is a really well-established risk gene, but usually when we talk about HOXB13 with regards to prostate cancer, we're talking about the G84E variant, which is only found in European ancestry men. But there's actually a number of other variants in HOXB13 that are population-specific and thought to be associated with prostate cancer risks, such as this particular variant that's only found in Japanese populations. There's one that's in Chinese populations. And then more recently, there's been this variant reported in African ancestry populations. So this study was done a few years ago now, where they did exome sequencing of HOXB13 in 46 early onset prostate cancer cases, and they identified 3 men who were carrying this variant. They weren't really able to do many statistical analyses because it was a case-only study and they only had 3 men carrying this variant. And this is an exceedingly rare variant, but it's a stop-loss deletion, so it has some potential biological relevance. So we thought we would follow up this variant in our large genetic association study data to see if we could actually calculate an association between this variant and prostate cancer risk. So we tried to genetically impute this variant into our GWAS data, and we did that using 2 different imputation reference panels. One was the 1,000 genomes reference panel, where there's about 2,500 participants in the reference data, and 3 of them are carriers for this particular variant. And the other one was TopMed, where there's about 97,000 participants in the reference data and 126 variants of this variant, carriers of this variant. You can see that the info scores for each of the arrays that we did the imputation in were really good for TopMed when we did the imputation with TopMed. And they're also pretty good when we use 1,000 genomes, but a little bit lower. But we found that the results were perfectly discordant. The carriers identified by one were not carriers in the other. So we did confirmatory genotyping with 1,500 men to determine which of these reference panels is actually working. And we found that the TopMed imputation, where we have a much larger number of participants and carriers, did really good. It was almost perfectly correlated, whereas 1,000 genomes imputation had a really poor correlation. So this also just goes to show how important it is to have diverse populations in these reference panels if we want to study rare variants that are potentially population specific. So once we had confidence in our imputation of this particular variant, we went ahead and looked to see if it was associated with prostate cancer risk. And we saw that men who carried 1 copy of this risk allele had about 2.4-fold increased odds of prostate cancer. But when we look at more aggressive disease, we see that the odds ratios get bigger and bigger. It goes all the way up to an odds ratio of 5 for men who carry this prostate cancer with the outcome of metastatic disease. So men carrying this variant have 5-fold increased odds of metastatic prostate cancer, suggesting that this variant is associated with overall risk and also really aggressive disease as well. And then the last thing that we did with this variant was to see how it's distributed across different African ancestry populations, which is plotted on this graph here. And you can see that the carriers, which are indicated by the black triangle, you can see that they all tend to fall around other participants who have West African ancestry. We don't see any instances of carriers among men who have North African ancestry, East African, or South African. It's really specific to men with West African ancestry, which has important implications for Black men in the US who tend to have more West African ancestry. So to summarize this section, could population-specific variants improve risk-stratified screening? So that this is the NCCN guidelines, and it's a lot of text. I don't want you to read all of it, but I've just tried to highlight some of the important takeaways for their guidelines on early prostate cancer detection. So here they recommend that men who are known to carry one of the BRCA2 pathogenic variants are recommended for screening starting at age 40. And men who carry other germline variants are recommended to start to consider screening at age 40 and to consider screening annually rather than every 2 years. But here they're really focusing specifically on rare pathogenic variants. They're not really thinking about common variants or these non-coding variants like we've been talking about in some of these examples. So expanding these guidelines beyond rare pathogenic variants could prove to be beneficial, especially if we're considering this 8q24 variant that is associated with increased risk of aggressive prostate cancer in men of African ancestry. And then second, they do mention HOXP13 in this guideline, but in the discussion they're really focused on the Scandinavian or European ancestry population-specific variant, rather than discussing the other variants that we know to exist in other populations. And so it'll be important to really broaden this to be a little bit more inclusive of the populations that could be impacted by genetic testing. So for the third and last section, I'm going to talk about some work we've done to combine polygenic risk scores and rare genetic variants. But since I haven't talked too much about rare genetic variants yet, I just wanted to give you a little bit more context to have a better understanding of how rare variants contribute to risk of prostate cancer and aggressive prostate cancer. So this is a study that was done a few years ago now, and it's a study in a sample of African ancestry men. And here they're showing that for these particular genes, BRCA2, ATM, PALB2, and MBN, there's some evidence of carriers of pathogenic variants in these genes having increased risk of overall prostate cancer and particularly higher risk or metastatic prostate cancer. Similar findings have also been done in European ancestry men. But one thing that I want to point out is looking at this particular figure from this paper from a few years ago. If you look across different labs where they have prostate cancer gene panels, you can see that there's often a pretty different set of genes that might be tested by that particular lab. And this is partly because there hasn't been the sequencing studies that have been done that are needed to look at the effect of rare variants on prostate cancer risk have tended to be pretty small. Sequencing is a really expensive technology, so the sample sizes to date haven't been huge, but that also means that the statistical evidence for any of these genes hasn't been very strong or consistent, depending on the gene that we're looking at. So we wanted to take a closer look at this, and we just had this paper that was published maybe just a couple weeks ago now, where we did exome sequencing in 17,000 men. This is a case-only study. Half of them had non-aggressive disease and half of them had aggressive disease. And of those with aggressive disease, most of them died from prostate cancer, and about 2,400 had metastatic prostate cancer. So we took a closer look. We did a lot of things in this paper, but one of the things we did was to take a closer look at the genes that are recommended or that are included in some of these gene panels, at least based on this study here. And also looking at some other candidate genes as well for prostate cancer. But we found that of those that are included in gene panels, we see pretty strong evidence for BRCA2, MSH2, ATM, and NBN. And we see some evidence for RAD51D and TP53 as well. But another important question is which ones do we really see no evidence for or very little evidence for? And of those that are included in gene panels, some of the ones that we had particularly low evidence for were RAD51C, MSH6, PMS2, HOXB13, BRIP1, and EpCAM. And you'll remember that we have talked about HOXB13. This is looking only in a European ancestry data set, so it's really thinking about the European ancestry specific variant, which hasn't in previous studies been shown to be associated with risk of aggressive disease specifically. And then when we also look at we also found that of the genes that are either in gene panels or have some evidence of being candidate prostate cancer genes, we found that about 11 of them had pretty good evidence in our study, and those are the ones that are highlighted in blue here. And we found that the carrier frequencies for these genes, rare variants in these genes, range from about 2.3 in men with non-aggressive disease up to about 7% in men with metastatic disease. And then when we look at family history, the differences aren't too different. The frequency is a little bit higher in men who are family history positive, but it's also pretty high in men who are family history negative. And I just want to highlight here that, if we have 2.3% of non-aggressive cases carrying these variants, that could suggest that these non-aggressive cases might be particularly important to follow up and see if they end up developing more aggressive disease as time goes on. And then with family history, a lot of the guidelines are often targeted around men who are family history positive. But there are a lot of carriers among the men who are family history negative, and that could be because a lot of men just might not know their family history that well. Maybe their relatives didn't live long enough to develop prostate cancer. So I think broadening the guidelines to not be too limited about family history would probably be beneficial as well. We also found that among carriers of rare variants across any of these 11 genes, that they had a younger age at diagnosis, about 2 years younger on average. And if we look at BRCA2 alone, carriers had about a 1 year younger age at diagnosis of non-aggressive disease, prostate cancer, and about a 4 year younger age at metastatic disease. And these findings are fairly consistent with previous studies as well. Okay, so that is kind of my short spiel on rare variants, but now we'll talk about integrating rare variants and polygenic risk scores. So we did this study a couple years ago in European ancestry men to try to evaluate what is the combined effect of rare variants and common variants as measured by polygenic risk score. So here we're showing the odds of prostate cancer on the y-axis. Let me see if I can get the mouse to come up again. In carriers, which is shown by the bright blue, and in non-carriers, which is shown by the dark blue. And you can see on the x-axis we have the different PRS categories. You can see that the odds of prostate cancer are highly dependent on what an individual's polygenic risk score is. There's a pretty wide range among carriers and non-carriers. And in particular, you can see that the odds of prostate cancer in non-carriers with high PRS is about the same as what it is in carriers who have average PRS. And the highest odds of prostate cancer, as we would expect, is among carriers who also have high PRS. When we look at absolute risk of prostate cancer by age 85 among carriers, it's about 26%, and it's about 11% in non-carriers by age 85. But if you integrate in information about a polygenic risk score, you can see that these absolute risks actually have a pretty wide range, ranging from about 2% up to about 30% in non-carriers, depending on what their polygenic risk score is. And then among carriers, those absolute risks range from about maybe 10% up to about 55%, again, depending on what their polygenic risk score is. So this was looking at a large sample of European ancestry men, but we also recently completed this study in a large sample or not as large of a sample of African ancestry men. The sample, because it was a little bit smaller, we look at these PRS tertiles instead of deciles like we did in the previous study. But the results are pretty similar. Here we see that among non-carriers with high PRS, their odds of prostate cancer is pretty similar to carriers who have low PRS. And then when we look at absolute risk of prostate cancer among carriers by age 85, it's about 48% and about 15% in non-carriers. But again, those vary widely based on what a man's polygenic risk score is, ranging from about 5% in non-carriers up to about 20%, depending on what their PRS is. And then In carriers, it ranges from about 10% all the way up to about 50%, depending on what their PRS is. So both of these previous analyses that I'm showing have focused on just overall prostate cancer risk. But in this sample of African ancestry men, we also had a number of men with metastatic prostate cancer. So we were able to see if this is true for metastatic prostate cancer as well. And you can see the results look really similar. The odds of prostate cancer in carriers is bigger than what we previously observed because carrying these rare pathogenic variants is often associated with more aggressive disease. So we expect to see that. But the results are fairly similar otherwise. And you can see that among carriers, the absolute risks of prostate cancer really depend on what their polygenic risk score is. So using this information in conjunction would likely improve predictive models of prostate cancer risk. So in summary, I hope one of the main takeaways is that it's crucial to include diverse populations in genetic investigations. This can improve risk models across populations and uncover novel biology. This can also enable the identification of population-specific variants. Our findings also suggest that genetics could be a way to reduce health disparities through improved screening and disease management. We found that the multi-ancestry PRS is a valid predictor of prostate cancer, and it has some evidence of clinical utility. We found that it's highly replicated across diverse populations, and it's somewhat predictive of aggressive disease and risk of upgrading. And we also found that there are some African ancestry variants that contribute to risk of aggressive disease, and that the influence of rare pathogenic variants on overall and aggressive prostate cancer varies based on polygenic risk scores. Some of the next important steps of this work are to further increase the diversity of participants included in discovery GWAS so that we can identify more population-specific variants and improve polygenic risk scores. And then it'll also be really important to evaluate the utility of polygenic risk scores and high-impact variants in clinical settings across diverse populations so that we can really understand what is the best way to integrate this information into screening, if we should be integrating it at all. So there's a couple examples of ongoing studies that are doing precisely this. One of them is the eMERGE network. They're prospectively returning polygenic risk scores for 10 different conditions, one of which includes prostate cancer, to 25,000 diverse participants. And the goals of that study are to assess patient understanding of genome-informed risk and the uptake of recommendations for disease management, as well as the impact on clinical outcomes. Another example is the GENOVA study, which is prospectively returning risk for 5 conditions, one of which is prostate cancer again, to over 1,000 diverse participants. And here they're going to assess the impact on clinical management, detection of undiagnosed cases, and the primary care physician knowledge and beliefs on polygenic risk scores. And both of these studies are really important in just understanding how can we actually use this clinically and how would it be perceived by participants and used by clinicians. But for prostate cancer in particular, it can take a really long time to develop the outcomes that we really need to know about, such as lethal or fatal prostate cancer. So there's still going to be a bit of work to do to really comprehensively understand how we can, what the impact of this information is on long-term outcomes. And then last, a study that we're working on here at Fred Hutch and UW is to enroll a new cohort of men who are recently diagnosed with prostate cancer across the state of Washington. And here we're going to limit the enrollment to non-white participants to try to build a really diverse cohort. And we'll follow them longitudinally and collect genetic information from them at baseline to see if there are genetic risk factors, either previously established ones or new ones that we identify in this study, that are associated with risk of progressing from perhaps a less aggressive disease to a more aggressive disease. We'll also enroll their male relatives with or without prostate cancer and follow them longitudinally to see either how their prostate cancer changes or if they develop prostate cancer over time. And this, because it'll be longitudinal, we'll have questionnaires about every 2 years. And at each of these, for each of these questionnaires, we'll ask them questions about their sociodemographics, their personal and family history of cancer. And then we'll ask if there have been any changes to their prostate cancer status or treatment or care. And then we'll have the saliva samples so that it's a really low barrier to participating, which we'll use to generate the genetic data. And then in addition to asking these questions about genetics, we'll also be able to ask, because we have the questionnaire and we can really flexibly include whatever people are interested to know about, we can ask if there are lifestyle and social factors that could also be related to disease progression, and if there's potentially interactions between these different lifestyle factors and genetic risk of prostate cancer. So thank you again for your attention today, and the work that I present today is really only possible due to a huge collaborative effort across many institutes and investigators and sources of funding. So thanks a lot to all the people who contributed this over the years, and thank you for your attention and for inviting me to give this talk today and share this work with you. Yeah.
Thank you. That was a wonderful talk. I wonder, do you think, or do you have any evidence of the idea that there could be genotypes that make other biomarkers like PSA either better or worse?
Yeah, that's a great question. So there is definitely evidence of that. So there was a PSA GWAS that was published. I mean, there have been a few over the years, but there was one that was just recently published in Nature Medicine. I think it was this year that it was published. And they found, I think, about 180 or so variants that are associated with PSA. And one of the interesting things that they found in that study was when they look at our prostate cancer polygenic risk score and account for the genetic variation of PSA, that the polygenic risk score actually does better. It becomes a better predictor of aggressive prostate cancer than it previously is. And in the new paper that we have that was just accepted at Nature Genetics with the larger GWAS of over 900,000 men, where we have this new PRS of 451 variants, we tried excluding any of those 451 variants that overlapped with that PSA GWAS, and there ended up being 51 variants that were also associated with PSA. And we found that the PRS became a better predictor of aggressive versus non-aggressive prostate cancer when we did that. So there's definitely some genetic variation of PSA that is kind of somehow entangled in everything, and we probably need to do a lot more work to better understand how to best tease them apart, because it seems like it's kind of, it's sort of a limiting factor that we could probably do better if we better understand the genetic contribution to PSA. Yeah.
And then one follow-up question. Are there, I buy from your data that there are genotypes that are associated with aggressive prostate cancer. Are there those genotypes that are associated with the exact opposite, that prostate cancer that will never kill the person, or that person will get prostate cancer, but it will never be dangerous? That's
a good question. I don't know if we have great evidence for that, because once you have prostate cancer, a lot of what happens afterwards might not be so genetically driven, because there are a lot of other factors that play into it, like what's your health care like, things like that. So I think there is evidence of some variants that may seem to be protective against more aggressive prostate cancer, but I think at that point there's probably a lot of other factors at play also.
This is just kind of a naive question, but how much overlap is there between these sort of germline variation that you see and somatic changes that occur in prostate?
That's a good question. I think Colin would probably be better at asking and addressing those questions, because I don't do as much with. Yeah, I mean, I think it's a really interesting question, and something that I would definitely like to get into more to better understand, like the overlap and what happens when you have both, when you have somatic and germline, and but I don't know in particular, like if having a germline factor makes you more likely to have the same one in the tumor as well. I mean, I don't know, maybe Colin probably is more familiar with that literature, but.
What we do know on that topic, not so much in prostate cancer, but in lung cancer, we know that, for example, East Asian ancestry is strongly associated with certain somatic mutations. So we know that in principle, what you're getting at exists, and there's every reason to think it's going to be true for prostate, but I don't know of any data on it yet either in prostate.
Yeah, that's an interesting question. Yeah. Ooh, yeah.
We have a question from one of our online attendees. Did you control for social determinants of health in your genetic analyses in different genetic backgrounds? Are your findings genetic or something else?
Yeah, that's a good question, and I think it's something that's kind of evolving in the field right now, how we think the best way to incorporate social determinants of health is. So one of the really nice things about germline genetics is that there are very few things that actually will change your germline genetic risk factors because it's inherited at birth. It doesn't change throughout your lifetime. So in terms of being like a true confounder, a social determinant of health is unlikely because it's unlikely that a social determinant of health would impact your germline genetic variants. But I think it is very likely that there is some effect modification or mediation that's happening through these social determinants of health that we haven't explored. I don't think there have really been many studies across genetic epidemiology in general that have addressed those questions, but it's very hot topic right now, and I think we'll start to see a lot more in the coming years. And that's something that we're interested in too, and thinking about how we can do these studies, and studies like All of Us, where they are collecting a lot of social determinants of health and genetic information. It's something we want to see, like, does the effect of the polygenic risk score on prostate cancer risk depend on someone's social determinants of health? So I think we'll see a lot more of that in coming years. Yeah, I guess too, there is some evidence of lifestyle factors being more or less important depending on like your polygenic risk score. For instance, in men who have really high polygenic risk scores and higher risk of prostate cancer, there have been some studies that have shown that having a healthy lifestyle is even more beneficial to those men. So there kind of is like a way to offset your risk. And that was looking at risk of like lethal disease in particular. And I think for prostate cancer, it's often a different question of overall prostate cancer versus lethal because the risk factors are very different. But yeah.
I wanted to extend the question. I understand there aren't known protectors for metastatic disease or protection, but what about protection from prostate cancer in the first place?
Yeah, for genetic risk factors, I mean, there certainly are. It kind of just depends on how you frame it though, because there's 2 alleles for any given variant, and it's either, if it's associated with prostate cancer risk, one of them increases risk and the other one decreases it. So it kind of just depends on how you look at it. So you could build like a predictive or a protective polygenic risk score and just think of it, but it would be the same variants. But there are certainly a lot of instances where the most common allele that's like present in 98% of men is actually protective against prostate cancer. Wait, is that right? Yeah, sorry. Yeah, you can look at it both ways, but there are instances where usually we're expecting the rare variant to be the risk increasing one, but there are instances where the rare variant is protective against prostate cancer. There are a lot of instances like that. And we also have seen in some of our sequencing studies, some findings that we have a really hard time interpreting. I don't know if I can pull it up quickly enough, but when we look at these rare variants, we're just looking at pathogenic variants. We would expect pathogenic variants to be associated with increased risk of any disease, but for instance here, there's this let me see if I have the mouse. This NEAL1, there is a p-value less than 0.05 here, where carrying pathogenic variants in NEAL1 is protective against aggressive and metastatic prostate cancer versus non-aggressive disease. And it's been really hard for us to know how to interpret that because it's not what we expect to see, but it's something that we have seen a few times. But it may not be replicated well. Like maybe this is just a one-off finding, so yeah.
I have a pathologist-centric follow-up to what Mark said, which is that there's some evidence, and it's spotty based on autopsy studies, that essentially everyone who has a prostate gets prostate cancer. So it would be difficult to answer Mark's question without autopsy at the end to prove that someone who didn't get diagnosed with prostate cancer didn't actually have prostate cancer.
Well,
are there autopsies in the studies that you're putting together?
This one is a case-only study, so it's comparing aggressive to non-aggressive. So they all have prostate cancer for this particular instance. But I guess like when thinking about the GWAS studies, when we're doing cases versus controls, I'm sure that there's a lot of misclassification because most men will end up developing prostate cancer at some point in their life, but it may not be clinically significant or detected. But it's a good question. I think, it kind of also goes to show that even with that misclassification, we're still able to detect a lot of variants that are strongly associated with prostate cancer risk. and they're very predictive in the end. But we don't really want to identify all of those clinically insignificant prostate cancer cases. We really want to be comparing the more clinically significant one to men who don't have prostate cancer. So, yeah.
So maybe this is just the same question as the one about social determinants of health, but I was wondering about interactions with other environmental factors that are strongly correlated with geography. So, how optimistic are you that a polygenic risk score calculated in North American populations would actually be predictive in very different environments? Like if you look at, men of Asian ancestry in Puget Sound, can you use the same polygenic risk scores if you go to Japan? And what is your sort of intuition about the interaction between variants and environmental factors?
Yeah, it's a good question. So in our GWAS studies, we do include a lot of international studies. So we include, for instance, like Biobank Japan is one of the studies that'll be in the newer GWAS that's published. And we have a huge number of men from North America, but also a lot from the UK, a lot from Africa. We don't have as many from South America, but we do have a lot of international studies that contribute to it. And when we look stratified by country, we do see that it works pretty well across different countries. But one thing that's really challenging is that the mean PRS, like if you look at the raw polygenic risk score, the distribution, it does vary between different ancestries and different, like even within like African ancestry, if you look at different countries, there's huge variation. So I think that kind of means that it's really hard to actually interpret what does like if you have a polygenic risk score of 23, what does that mean? Who do you compare it to? So this is like a big problem, I think, beyond prostate cancer, just in polygenic risk scores in general. How are we going to actually interpret what they mean? Because who is it relative to? Because that will make a big difference in whether you're considered to have high or low risk. And then for rare variants, we've also been able to include a lot of international studies. And I think one interesting example for rare variants there are some fairly reasonable differences in frequencies between countries. And for instance, for BRCA2, we saw a really strong association for risk of aggressive disease in men from the UK. It might just be that like when they sent us the samples, they sent really aggressive samples. So maybe that's part of it. But we see really weak associations, like almost null, in men from Finland, which is consistent with some previous, it is consistent with some previous studies that BRCA2 isn't as important of a risk factor for some reason in Finland. But there's definitely differences between countries and regions that complicate things a little bit. Yeah.
Well, I was thinking about your observations of a connection between PSA and both the risk and the genetic factors. Is there a possibility that we should have a genetic reference range, genetically determined reference range for PSA?
Yeah, I think something like that would be great. And I think that's something, the people who did the PSA GWAS that they published, they came up with a polygenic risk score for PSA. And they used that polygenic risk score to genetically adjust the PSA that you do in clinic. And I think doing something like that could be really helpful because, if we just use PSA, it tends to overdiagnose men. But it seems like if you do this genetic adjustment for PSA, that it might help identify the men who actually have risk of or have aggressive disease. So, yeah, I think something like that would be great. But yeah, I mean, I haven't been directly involved in that research, so I think they probably have thought through those details a little bit more than I have. But I think it's a great idea. Was there a question back there?
There are studies that are considering genetic modeling of the genes which are variant so that to reduce the risk of prostate cancer.
Sorry, can you say that again?
There are studies that are considering genetic remodeling of the variant genes that triggers prostate cancer.
Oh, genetic, like editing, is that what you said?
Genetic remodeling.
Remotely? Remodeling
Yes.
Of the variants that are
causing prostate cancer.
Oh, when you say genetic remodeling, is that like genetic editing, kind of? Okay. Not that I'm aware of, but maybe, because I haven't been in that space at all, but it's an interesting question. I don't know, it kind of, yeah, I don't know. Not that I'm aware of, but maybe, yeah. Maybe I haven't been involved in those discussions or that space of research. Yeah.
Thanks, Bertie, again. Great job.
Thank you.